Hello, everyone, and thank you for coming to my lawn talk. My name is Andrew Dancer, and I'll be presenting an exciting use for optogenetics that will help us to understand better the function of specific brain regions. This is in the hopes that new understanding will give rise to new treatments for common anxiety and fear-based disorders. Before we hop into the experiment, I'll be covering the background of what optogenetics is, since it is such a new field and goes largely unnoticed in many common pools. I'll also briefly cover the benefits of optogenetics and what it boasts over more common treatments today, to serve as examples of how treatment may compete with or even complement existing treatments. When it's time to cover the experiment, I'll go over the hypothesis describing what they expected to find during this experiment and where they may have been a little bit wrong in their assumptions. I will cover from some of the tasks carried throughout the experiment, as it had many different parts looking for many different interactions. Some will cover responses in live mice, while others will involve the processing and examination of the mouse's brain post-test. Then I will cover the results that I found most important through the course of the experiment, what they mean, and the promise they show for future research into optogenetics. First, what is optogenetics? Breaking the word apart, we have opto, meaning light, and genetics, meaning to do with the genetic material of an organism. With this understanding, we know that optogenetics is a field of biology that aims to use both light and genetics to achieve a goal. When broken down like this, it's fairly simple to understand the goal of optogenetics, although the process of optogenetics is not as clean as this can make it out to sound. First, a viral vector is prepared, seen as right here, and it's used to infect cells of varying specificity based on the goals of the procedure. This viral vector carries the genetics for a specific light-sensitive protein. Today, it's often a rhodopsin of some kind. Those are very popular in today's optogenetic examinations. These proteins are selected for their ability both to interact with specific cell functions and because their geometry and therefore function can be altered by the addition of certain wavelengths of light. Many different proteins can be chosen at once to affect many different cell types and be individually controlled fairly reliably with optogenetic implants, which, coming to the implants, they are probably the most unsettling and intrusive part of the procedure, often seen in labs today, is the addition of fiber optic wires that flow through the skull and into the desired region of the brain in order to shine light directly onto a target area reliably. Although more mobile and reasonable methods are not so outlandish to design. Designs such as LEDs that use batteries similar to those used in today's pacemakers could be something as an example. The world of psychological disorders is a messy one, with a wide variety of treatments and an even wider variety of disorders. For fear and anxiety-based disorders, pharmaceuticals and therapy are common treatments, but see several unfortunate limitations. Pharmaceuticals require trials of chemical after chemical after chemical in order to find the correct molecular geometry to produce the desired effect. And it can be very difficult to give the body a molecule in its common form. Often an intermediary molecule is found, and production or harvest method of that molecule must be discovered, and even then, the struggles are not done. Side effects can be extreme and difficult to predict. Delivery methods can unintentionally cause unintended alterations in undesired cells. and individual medical history can be a nightmare in changing a person's susceptibility to drugs or even the effects of those drugs entirely, as seen in things like antidepressants, drugs that can even strengthen connections and effects that they originally sought to dampen. All of those problems can make finding, researching, producing, and approving new pharmaceuticals extremely time and cost intensive. And for some disorders, simply we don't have the time or the money to spend on them. Therapy comes into both similar and unique problems. With so many approaches to therapy, it can take some time for proper method to be found. And once a therapist is found that whose method agrees with the treatment plan, it often takes a significant amount of time for the relationship to build and progress to be made. Therapy also sees spotted record for effectiveness after sessions have stopped, with individuals fighting a relapse later on. The improvement seen by therapeutic extinction of negative memories is not a flawless fix, and the same connections within the brain still exist, leaving the potential for problems to resurface. Anxiety and fear-based disorders are no joke when it comes to numbers. Millions of people worldwide are affected by these disorders, with the National Center for Biotechnology Information, otherwise known as NCBI, reported that upwards of 30% of people may suffer from anxiety-based disorders within their lifetimes, with another 4% affected by fear-based disorders. Now, for anyone who's desensitized to statistics, that translates to over 2 billion people in just today's population who will experience an anxiety-based disorder within their lifetime, while over 300 million will suffer from a fear-based disorder. Now, for anyone desensitized to those large numbers, that translates to a room of just 25 people, I might add, smaller than my average high school class. One of those 25 people will suffer from a fear-based disorder, while 8 will suffer from anxiety-based disorders. And this is just a normal average room of people. We found that these disorders share a common root in memory through previous research, seeing patterns in neural connections affecting how memories are stored, retrieved, altered, and even erased. Some key terms to understand what is happening with memory in relation to the neurons, and in turn, the memories associated with those neurons, are as follows. Acquisition is the creation of a memory's connections that lead to it becoming an established part of the neural structure. This is experienced when an organism is introduced to a stimulus for the first time. Consolidation is a time frame of susceptibility that memories have, where the neural connections are open to alteration, natural or artificial. Reconsolidation is when a memory is put back into a state of susceptibility to alterations and is often involved in the retrieval of memories, as memories can change the more and more we retrieve them. Extinction, however, it is important, is not what the name makes it out to be. We actually named this process before we fully understood what was going on. Originally, we thought that this was a process of unlearning and assumed that the neural connections would be degraded or destroyed. But in reality, the neurons are actually creating alternate routes, growing more in order to learn to avoid that original pathway and the memory associated with it. And this ends up being a way of the brain working around its problems rather than trying to just erase them entirely. This allows for, say, if that same fear condition should show up again, the organism would still know what to do. Because of this, extinction should be seen as a process of growth, not of destruction. Through previous research, we know that the hippocampal dentate gyrus, which will from now on be referred to as the DG, is a major locus of neuroplasticity. It can be manipulated to inhibit the formation of memories or to interfere with the formation in such a way that the connections are muddled and association becomes highly generalized, displaying fear responses in response to stimuli not very similar to the original. This will be the main focus of the experiment, although this region of the brain also sees a strange ability to inhibit the effectiveness of antidepressants. It's suspected as being a pattern separating center for memories associated with fear and anxiety that can cause disruption during memory acquisition of negative stimuli, can lead to overgeneralization of the stimuli and development of an anxiety or fear-based disorder around that memory. From the original research looking into this region of the brain, it was concluded that the DG plays a role in memory acquisition, but not in memory retrieval, and subsequently possible memory modification to existing memories. However, this was disputed when the next experiment concluded that the DG played a role in both. This became the focus of today's experiment, with each task carried out having the intention of answering this debate. The expected result was that the suppression of the DG via optogenetic manipulation would interfere with the neural pathways and cause a decrease in fear response. This turned out to not be entirely correct in some surprising and unexpected ways. The hypothesis was that the DG is important both to acquisition and retrieval because they disagreed that the DG was not important at all to retrieval, concluding that it must play some role in the retrieval of memories despite its shown inability to prevent recollection and retrieval when inhibited. The subjects used for this experiment were male laboratory mice. They were kept in extremely controlled environments with identical temperatures, areas, lighting, even as specific as the scent of the testing areas. When the mice were between 8 and 10 weeks old, they underwent optogenetic procedures to be injected with the viral vector in 2 locations bilaterally along the spine. The viral vector targeted DG neurons, giving the infected cells the genetic code to produce the protein EGFP, also known as halorhodopsin, a light-activated chloride pump capable of impairing neuron function. Infection was mostly exact, with minimal gene expression found in small numbers of other areas throughout the brain. During the same surgery, those mice had identical incisions made in their skulls to give access to the DG. Fiber optic wires were run through the brain to the DG in order to shine light through to the cells to activate the halorhodopsin produced in those target cells. After being allowed to habituate to the implants for 2 to 3 weeks and being allowed to habituate to being handled for an additional 4 days after that, testing began. Novel experiment exposure was performed in 4 gray open areas with walls, completely opaque, lit by uniform incandescent bulbs. Mice were transported directly from the holding area to the testing room, connected to fiber optic patch cables, and immediately placed into a novel open field apparatus for 15 minutes, after which they were returned to their cage. 90 minutes later, the mice were killed, their brains were dissected, and they were prepared for sectioning. During fear conditioning, mice were transported to holding area 1 hour prior, then introduced to the testing area. Containing nothing but walls and a steel gridded floor, after 180 seconds, they were shocked once, then removed from the testing area after another 30 seconds. The mice were split into groups based on the presence of genetic alterations and the presence of light being actively shown through the optical fibers. Extinction testing occurred once daily for mice for 5 days following the testing, consisting of 5 minutes in the testing area with no shock. 5 tests were given out until later in the experiment, when 2 additional test days were added in order to determine if the continued inhibition of the DG during extinction gave a different result. Brains were then cryogenically preserved after dissection and sectioned into via a very complex and specific chemical process that will not be detailed here. The target site in the brain can be seen in the image on the left, as well as the comparisons on the right, showing examples of mice labeled appropriately. The types of mice that will be referred to are DG halo and DG GFP mice. Halo mice are ones with halorhodopsin gene artificially added by the viral vector, while GFP mice have no such genetic modifications. A term called freeze rate was used to score the reactions of the mice during extinction testing based on a blind scoring of the mice's reactions throughout the tests, looking for sudden stopping of motion. DG impaired halo mice showed consistently higher freeze rates when observed in extinction testing. Testing seemed to support the original conclusion that the DG does not play a significant role in retrieval of memories, and could only be used to manipulate the connections of memories during acquisition. However, a simulation program that was also a part of this experiment, called Bacon, affectionately by these scientists, came to the aid of these researchers and helped them understand what the case may be for this DG experiment. No, the name is not a joke. It's really called Bacon. It's not quite what the acronym actually is, but I can't complain about Bacon. Bacon stands for Bayesian Context Fear Algorithm and is a simulation software used for predicting neural responses. This simulation suggests that perhaps the 2 studies, which originally showed conflicting results, may actually agree with each other. The disruption of the entire DG versus the disruption of even more specific cells within the DG may actually be what's causing this discrepancy in results, although more studies must be done to conclude this. It is believed that perhaps the inhibition of these specific neurons allow for the rest of the DG to aid in retrieval of other contextual information and avoid the generalized fear seen in some of the subjects, where they displayed fear responses in situations that were slightly similar, but not entirely similar to the original stimulus. What I would like for you all to take away from this is that optogenetics is an exciting new field, capable of breaking barriers never overcome by more traditional treatments. And the more research we can perform into understanding how to manipulate neural activity with the help of optogenetics, the more we'll be able to help the millions of people in need of our help. Pharmaceuticals and therapy are great tools, and there's good reason for their widespread acceptance and use. However, I don't believe they're reasonably fast enough to aid humanity's growing ailments, as these cases of fear and anxiety-based disorders continue to climb in shocking numbers. Being able to bypass common problems like the blood-brain barrier could prove invaluable in the treatments available. And with the help of optogenetics, I believe it's possible to help many more people in a much simpler fashion. Now, this may be a scary topic of discussion. The artificial manipulation of memories is a dystopian concept, often not seen far from its popular promulgator, Blade Runner. However, that's simply not the reality of the technology, and real investment into understanding it may allow us to effectively treat people with disorders related to memory. Your memories may not be Play-Doh to science like they are in Blade Runner, but they certainly are not written in stone, and we can use that to our advantage and to help people. Thank you for attending my long talk. I hope that you all have a wonderful rest of your day and that you enjoyed and learned something here. Thank you.